Department of Clinical Genetics of the Erasmus Medical Center is a large department, 250 people work there, and we combine clinical care, laboratory diagnostics and research. And that combination gives a very strong impact on the work we do. Most of our work focuses on neurological disorders, on congenital malformations, on cardiovascular diseases, and also very important is the prenatal diagnostics. The other thing which is unique for our department is that we have brought the functional genomics into our diagnostics.
We had one case which was particularly challenging. It is about a boy, 20 years old boy, with multiple congenital anomalies and developmental brain disorder. And there was not yet a syndrome describing the complex of his problems.
Nou, als je niet weet wat voor diagnose die heeft, dan weet je ook niet hoe die zich zal ontwikkelen. Dus je was altijd een beetje angstig. Wat zou er nog meer komen?
By whole exome sequencing, we identified a homozygote mutation in INTS1. We identified 2 more families with the same identical mutation. At that point, we were sure from the functional side and from the clinical side that the INTS1 mutation was the cause of the disease.
We waren echt heel erg verbaasd van, dat er nou toch iets gevonden is. Ook een opluchting, omdat eigenlijk weten we nu wat hij heeft. We hoeven niet meer bang te zijn dat er nog andere enge dingen nog bij komen en dat er weer iets ontdekt wordt.
The toolbox we have is getting bigger and bigger, so we can do far more than we could 10 years ago. But the data we get is enormously complex. For that you really need a large team who has expertise in many different fields. And that's why we set up these multidisciplinary expertise groups.
The knowledge on cardiovascular disorders is like a big black box, and we don't really know much about the cause and the prognosis of these disorders. Therefore, we use the input of all the specialists involved in the care for patients with cardiovascular disorders, like the cardiologist and the vascular and thoracic surgeons, the pathologist, the radiologist, and of course the geneticist.
For aortic aneurysm patients, it's helpful for us as a surgeon to know whether or not there is a genetic basis for their disease. Exact genetic diagnosis helps us in making the best choices for patients, whether or not they need a prophylactic operation to treat their aneurysm.
By getting the samples from the thoracic surgeon, we make RNA profiles of both healthy tissue and aneurysmal tissue. We can see new pathways that are involved in the etiology of aneurysm disease. Earlier we found mutations in the SMAD3 gene, and by identifying the gene we made a special approach for patients with SMAD3 mutation.
Finding genes is becoming more and more common, so we try to use our genetic data to come up with therapies.
We are looking for genes involved in Parkinson's disease and other movement disorders, and in this research we also found a gene involved in a disease termed paroxysmal exercise-induced dystonia.
This disease causes a defect in the metabolism of the amino acid valine, and we've been able to show by mass spectrometry elevation of the toxic metabolites that these mutations are disease-causing.
In the field of neurological disorders and movement disorders, there's very little you can treat at the moment, and this is one of the very first examples where a specific molecular alteration has led to the possibility of a specific therapy based on the molecular alteration.
The multidisciplinary expertise groups we have in our department bring together doctors and the researchers, and that gives you the possibility to do things which most labs cannot do.
I'm part of the gastrointestinal group, and our research actually aims at understanding the molecular mechanisms involved in the development of several congenital intestinal disorders. We use in vitro assays, and we also have an in vivo model, that is the zebrafish. For each prong, we have a transgenic line where all the enteric neurons are fluorescently labeled. The ultimate goal of our research is actually to come up with a therapy for each prong patients by developing inducible pluripotent stem cells from patient skin biopsies.
One of the teams, the prenatal diagnostic team, were one of the 3 labs which were selected to be involved in the non-invasive prenatal test study, for which we now are screening over 20,000 pregnant females.
We want to offer more in a genetic test. Ethicists fear that if we do this, that people will not think anymore about taking the test because it's so difficult to grasp. We need to determine whether this is true or not on a scientific basis. We are looking into a model which focuses more on the deliberation process, and we are building a scale in which we can measure deliberation. So far we have learned that the so-called unexpected findings, other than trisomy 21, 18 or 13, that people can handle them when the genetic counseling is done in a very appropriate way, and we are learning the most appropriate way to do this.
Potentially, the impact of our work can be enormous. Clearly, we are going towards a personal treatment based on your DNA profile. We also have to keep in mind that we gain a lot of knowledge, but what kind of information do people want? That's something we really should pay attention to.